Good morning, my dear students. So glad to be with you, your bio teacher, Wagdi Amin. This year, was so proud and glad to be with you for the first time. And today, finally, we are going to finish the last part in your course for second stage for bio. This is DNA and protein synthesis. At the beginning, I'm going to explain what is meant by protein synthesis. Protein, this is something every minute inside your is synthesized. What are the examples of protein? Protein may be your skin protein. Hair, this is protein. After you cut it, it grows once more. This is protein. The food inside your body, to be digested, it needs enzyme. Enzymes which digest food continuously, type of protein. The hormone which organize different vital processes inside your body, this is protein. If I'm going to explain what is a protein, this is your life. So, I'm going to classify the protein into two types. The first type of protein, we have structural protein. What is meant by structural protein? Protein enter in a structure of your body, like actin, which form the muscle, like keratin, like collagen. They are all different type of protein, but they work as a structural protein, enter in a specific structure inside your body. This is the structural protein. We have another type of protein, we call it regulatory protein. What are the regulatory protein? Something like enzymes, something like hormone, something like antibodies. Enzyme and hormone and antibodies, they organize different vital processes inside your body. And because they organize and regulate processes inside your body, we call them regulatory protein. Let's start to see what are the different type of protein, structure and regulatory, and I'm going to start with the structure protein. Type of protein, there is enormous variety of proteins that enter in the structure of body, and we are going to divide them into two main groups, which are the first, this is structure protein. They are protein enter in certain structure in living organism. Like what? The first, actin and amylin, and I think you hear them before, they enter in the structure of muscles and other contractile organ. Collagen, they form connective tissues like ligaments and tendon. Finally, keratin, they form protective coverings like skin, like hair, like hooves, horn, feathers, and others. The second type of protein, we call it regulatory protein. They regulate processes inside your body, like regulate different processes and vital activities in living organisms, like enzymes. They catalyze chemical reaction in the body of living organism. Antibody provide body with immunity against infection by foreign bodies. And finally, hormones and other substances enable the body to respond in a proper way, continuously changing internal and external environment. To explain what is the structure of protein exactly, I'm going to start at the beginning. And may you remember, you studied that the building unit of protein, something we call it amino acid. There are 20 different kinds of amino acid enter structure of protein inside your body. But What is the basic building unit for protein? The amino acid. What is the structure of amino acid? Let's start to see it. Amino acid, the protein composed of 20 types of amino acid that have the same basic structure. Amino acid join together through dehydration reaction, and between them they are bonded by peptide bond to form polypeptide polymer, and in turn it will form the protein. The difference between different proteins are due to, this is the reason for difference protein from each other. The difference in number, type, arrangement of amino acid in the polymer. Again, differences between protein are as follows: the number, type, and arrangement of amino acid. The number of polymers that form the protein. And number 3 and last one, reason for difference of protein, weak hydrogen bond that may give the protein molecule its special shape. Amino acid, as you know, this is the basic building unit of protein. Structure of amino acid will be as following. This will be carbon atom connected with COOH group, or we call it carboxyl group, and connected with hydrogen atom. And number 3, alkyl group, and the alkyl group which differ from an acid to another. What makes the amino acid different from each other? This is the alkyl group. Finally, this is amino group. So, this is the structure of amino acid. Its structure as following: carbon atom connected with carboxyl group connected with amino group, and hydrogen atom and alkyl group. And be careful, if you please, the alkyl group differ from amino acid to another. This what I told you. Alkyl group, or sometimes we call it terminal group, differs from amino acid to another. They are, or alkyl group or terminal group, found in 19 amino acid only. And there is glycine amino acid, the only amino acid that has a second hydrogen atom instead of alkyl. This is the shape of glycine. This is COH or HN2. Sorry, instead of R, we have another hydrogen. So the R doesn't exist here. Okay? This is the glycine structure. Ribonucleic acid. We have different type of nucleic acid. You know before, and you studied the DNA, and we have another nucleic acid, we call it RNA. DNA and RNA. So important to differentiate between them. What are differences between DNA and RNA? At the beginning, you're going to study some similarities, and after that, you're going to study differences. Similarities and the difference, this is what you need to understand. At the beginning, I'm going to tell you, both of them consist of nucleotide and contain nitrogenous bases. They are different. Both of them contain of nitrogenous base. And differences. DNA consists of double strand, while RNA single strand. DNA consists of 4 types of nitrogenous bases: A, T, C, G, while RNA consists of A, U, CG, U in RNA instead of T in DNA. The sugar which enter in DNA, we call it deoxyribose sugar. This is why DNA, deoxyribose sugar. While RNA, ribose sugar. What is the difference in the structure between this? Let's start to understand them. This is DNA and RNA. At the beginning, I told you we are going to make the similarities or understand similarities. Similarities between them. Both of them are formed long, unbranched chain of nucleotide. Both of them consist of nucleotide subunits. This is the first. The second, each nucleotide made up of, this is 5-carbon sugar or pentose sugar, nitrogenous base, and phosphate group. The nucleotide phosphate group is bonded to the 5th carbon atom of sugar, molecule of one nucleotide, and 3rd carbon atom of sugar of next nucleotide. I think you understand this well, to form the sugar phosphate backbone. What are the differences between them? The first, the type of pentose sugar. Inside your DNA, we have the oxyribose sugar, which contain one oxygen atom less than the ribose. Its structure C5H10O4, while ribose sugar, its structure C5H10O5. What is the difference? Something so small. The ribose or RNA O5, the oxyribose or DNA O4. This is the first difference. Nitrogenous base, this is A and G, A and G. Purines in both of them are the same. While purines, this is thymine in DNA replaced by uracil in RNA. This is cytosine, and cytosine the same. So you have only difference in nitrogenous base. T characterizes DNA, while U characterizes RNA. The number of strand, double-stranded molecule or 2 complementary strand, while RNA, this is just single strand of nucleotide, but may be double-stranded in some of its parts, like tRNA, as you are going to study. Site of its presence, sure DNA found in the nucleus, while RNA transcribed from DNA inside the nucleus, then transferred into cytoplasm. Stability, sure DNA obviously stable, it isn't hydrolyzed or isn't hydrolyzed, while RNA continuously destroyed and rebuilt. DNA, you have just only one type, while RNA, there are 3 different types, which are mRNA, we are called it messenger RNA, or RNA, we call it ribosomal RNA, and tRNA, we call it transfer RNA. So DNA only just one type, while RNA, there are 3 types. Types of ribonucleic acid. As I told you, this is messenger RNA or mRNA, ribosomal RNA, and its abbreviation rRNA. Finally, transfer RNA, or we call it tRNA. Let's start to study the different parts which form the machine which are going to synthesize protein. By the way, I need to copy paper. If I need to copy a paper, you're using copying machine. I'm going to put the paper, copy it, and produce thousands of copies in a few times. Exactly the same, this is what happened inside protein synthesis. There are 3 different parts which are mixed together and cooperate together during protein synthesis. What are the different parts? I'm going to start to tell you. The different part, this is messenger RNA and its strand. How we will produce this strand, I'm going to explain exactly how it is transcribed. And the second part, which form the protein, this is ribosomal RNA. What are its parts? I'm going to explain. Finally, transfer RNA or tRNA. They together cooperate to finally form the protein. I'm going to show you a small video to summarize our processes, and before I'm going to start, I'm going to see you to make you see this video. Let's start. This is the shape of cell, and inside the cell we have nucleus. Inside the nucleus you have many chromosomes. There are 23 pairs. And inside chromosome, DNA consists of histone and non-histone protein which organize the DNA inside the cell nucleus. And this is the beginning of your DNA for transcription and translation. It starts with enzyme, we call it RNA polymerase. RNA polymerase will bind to sequence. Sequence, we call it promoter, at which transcription process starts. What I'm going to start with now, this is process of mRNA transcription. By the way, we are going to prepare 3 parts of the machine. What are the 3 parts of machine of protein synthesis? The first part, mRNA. To form mRNA from DNA, it's a process of transcription. To form rRNA from DNA, this is transcription. To form tRNA from DNA, also process of transcription. And transcription process, these 3 processes, occurs inside the nucleus. Transcription process, whatever, for mRNA or tRNA or rRNA, all of them, they occurs inside the nucleus. After they start to combine together and work to synthesize protein, the name of process, translation. If you please be careful for this because it makes difference. Translation process to form a protein from mRNA, this is occurs inside cytoplasm. While transcription process occur inside the nucleus. Again, transcription to form NA from DNA takes place inside the nucleus, while translation to form the required protein, this takes place inside cytoplasm, and the name of process, translation. Let's start. The shape of DNA strand, and I'm trying to form this strand. We call it mRNA, and this is a process of transcription. This is messenger RNA. Sure, the messenger RNA consists of, at the beginning, a start codon, and at the end, a stop codon. A start and end. After it transcribed inside the nucleus, it moves to the cytoplasm. This is the shape of cytoplasm. And the protein synthesis takes place inside cytoplasm. This, the different parts which combine together to start the protein synthesis. The upper part, a part of machine, this is large ribosomal subunit. And the small part, this is a small ribosomal subunit. And this is the strand of mRNA. Let's start what happened. This is messenger RNA. This is the shape of tRNA carrying amino acid. This is the shape of tRNA or transfer RNA carrying amino acid. Okay? And it moves along the mRNA. Amino acid, amino acid, amino acid. They bind it together to form the required protein. And this is a process of protein synthesis takes place as you see. After protein is formed, some modification takes place on it to perform its function. Okay? This is the shape of video number 1. Protein synthesis translation. What is the translation? Another shape make you to understand. This is cell, and inside cell nucleus, and this is endoplasmic reticulum, if you remember. Something transcribed inside the nucleus, this is mRNA. mRNA starts its function by combining with part, we call it, this is tRNA synthesis. You have two sites. This is peptidyl and amino acid site in the large ribosomal subunit. Let's see it again. They combine together. ATP give energy for the process. This is the tRNA, which carry amino acid. tRNA, which carry amino acid. Now, the process of synthesizing protein starts, as you see. This is the first tRNA. It has anticodon, which may be bind with mRNA. Let's see it. This is initiation stage, the beginning. I'm going to make you understand something. This part of ribosome, and we call it a small ribosomal subunit. And this is the mRNA. And this is the third part, tRNA carry amino acid. Let's start how they work together. Small ribosomal subunit. And this is codon. Each codon consists of 3 nucleotides, like you see. This is ribosome and the small ribosomal subunit. The codon consists of 3. By the way, this is AUG. The codon for methionine amino acid. Any process of protein synthesis start with this codon, AUG. We call it a starch codon. And binded with a small ribosomal subunit. This is tRNA, transfer RNA, carrying amino acid, and it will be methionine. And we complete the machine with large ribosomal subunit. The process takes place as following. This is A site, This is B site. This is E site, exit. This is peptide, where the amino acid bind together. This is where the amino acid starts to enter to the large ribosomal subunit. E, this is the place where the amino acid and transfer RNA exit. Transfer RNA exit. While the amino acid bind with the second bind with the third bind with the fourth, as you are going to see. Let's start to see it. This is the initiation complex, ribosomal subunit, small and mRNA, and this is tRNA. This, another tRNA carrying the second amino acid. So, we bind the first amino acid with the second amino acid. And the ribosome move along the mRNA. Be careful for this. The ribosome moves, you see it? Moves along mRNA. I binded the first amino acid with the second. This is tRNA number 3, transfer RNA, carrying the third amino acid, and it enter to the process, binded with the third, the first with the second, with the third, and the tRNA are freed to pick up another amino acid. This is the fourth, this is the fifth transfer carrying this amino acid, and now we are trying to bind the amino acid together with each other. And this is the part of the last part which we reach stop codon. You have 3 types of stop codon. Maybe yuga or yag or ya. UGA, UAG, UAA. Yuga, yag, ya. There are 3 stop codon. When we reach them, the protein synthesis process is stopped. This is the shape of these 3 parts. After you reach stop codon, one of the 3, which may be yuga, UGA, yag, UAG, ya, UAA, the protein synthesis process is terminated. So the 3 parts are separated. What are the 3 parts? The large ribosomal subunit, the small ribosomal subunit, and the mRNA. Separate from each other because protein synthesis process terminated or stopped. This is the most difficult lesson inside your course, wishing that everything you understood it in a good way. So proud and so glad to be with you. Hope you are doing great. Pray, thank you for your time. Happy to see you once more till we